Welcome. My name is Nicole Zabo, and I am the Communications Coordinator at the Invasive Species Center, and I'll be your moderator for today's webinar titled "A Bacterial Immune System Can Now Help Identify Invasive Plants, Weeds, and Other Plant Pests." Before we begin, I would like to acknowledge that the Invasive Species Center honors the long history of the First Nation, Inuit, and Métis Nation peoples on the lands now known as Canada, and strives to show respect to their ancestors, their communities, and to them individually. We greatly appreciate the significance of the lands, waters, and all living things, and offer our gratitude to the Indigenous people for their care and teachings about our Earth. Our relationships with Indigenous communities are important, and we will continue to listen and learn how we can be in a good relationship with Indigenous peoples, the lands and waters, and all living things, and act accordingly. The Invasive Species Center respectfully acknowledges that our head office is located on the traditional lands of the Anishinaabe, the Batchewana, and Garden River First Nations, as well as the longtime settlement of the Métis people in the Robinson-Hurry Treaty area. The Invasive Species Center is a not-for-profit organization that mobilizes action against invasive species that harm the environment, economy, and society. The Invasive Species, or ISC, has many great invasive species resources on our website, including species profiles, best management practices, video resources, and more. So you can check us out at www.invasivespeciescenter.ca. You can also sign up on our homepage for our quarterly newsletter, biweekly media scans, and event invitations, which is where you can hear about upcoming webinars and keep up with invasive species news. The ISC has also launched an invasive species training program, which offers virtual, self-paced courses for anyone wanting to learn more about the impacts of invasive species in their workplaces or communities. We currently have 4 courses available, which focus on forest invasives, but we'll be releasing new content soon, so stay tuned for that by signing up to receive updates on new courses and further course details. For today's webinar, we are collaborating with the Greenbelt Foundation, and I'll tell you a little about Ontario's Greenbelt and its agricultural production in the next couple of slides. In 2005, legislation was passed to create Ontario's Greenbelt, which is the world's largest greenbelt. It protects 2 million acres of land, including farmland, forests, wetlands, rivers, and lakes, and notably it includes the Niagara Escarpment and Oak Ridges Marine. And all this is supported by the Greenbelt Foundation's investment and stewardship. Ontario's Greenbelt is a working landscape that contains over 4,000 farms, and where actually 40% of the total land area of the Greenbelt was used for agricultural purposes. Fertile soils and unique climate conditions allow Greenbelt farmers to grow crops that can't be found commercially anywhere else in Ontario. And it's home to Ontario's only 2 specialty crop areas, the Holland Marsh and the Niagara Tender Fruit and Grape area. Greenbelt farms produce a large diversity of crops and are a significant supplier of Ontario's fruit and vegetables. There are a couple other points I would like to mention today before we get started with the webinar. So there will be questions, time for questions at the end of the webinar. So if you do have a question at any time, you can type it in the question box and I will read it to our presenter after the webinar. If you're having technical difficulties at any time, please type them in the chat box or respond to the email found in your registration, and we will resolve it for you. We also have enabled closed captioning, so if you would like to follow along that way, you can turn that on with the closed caption button on your taskbar. Lastly, there will be a brief survey coming out following the webinar. If you could take some time to fill it out, we would really appreciate it. Sorry, just one moment. I just have a little bit of a cold. Go ahead. Sorry about that. So without further ado, today's webinar is titled A Bacterial Immune System That Can Now Help Identify Invasive Plants, Weeds, and Other Plant Pests. And I'm pleased to introduce our speaker, Dr. Leonardo Galindo González. So Dr. Galindo González did his undergraduate studies in biology at the National University in Bogota, Colombia. He conducted thesis research in the evolution of transposable elements in common bean at the International Center for Tropical Agriculture in Cali, Colombia. He then moved to Canada to do his Master's of Science at the University of Alberta, working in dormancy of white spruce using omics. After completing his master's degree, he pursued a PhD studying the molecular responses of flax to Fusarium oxysporum. During his PhD, he also developed techniques to evaluate mutations using next-generation sequencing. Sorry, one moment, I'm just gonna cough. And standardized transposon-based. During his PhD, he also worked on standardizing transposon-based molecular markers for diversity studies in flax cultivars. As a postdoc and research associate, he studied the pathology of clubroot in canola and tan spot of wheat. As a research scientist now, he leads the Molecular Identification Research Lab, and he uses innovative methodologies to find and use novel molecular fingerprints that allow us to identify regulated, invasive, and weedy species to support early detection, prevention of introduction, and risk assessment of noxious plant species. And with that, I'd like to thank Dr. Galindo González for joining us today. I'll stop sharing my screen and then pass it over to him.
Thank you very much, Nicole. I'm gonna share my screen. Just give me 1 second. Trying to see if hold 1 second. And can you see my screen?
Yes, it looks great. Thanks, Leo.
Okay. So 1 second. All right. Are we good?
Yes.
Can you guys hear me okay?
Yep, all looks great.
Excellent. Okay. I'm just going to use this pointer here. Let's see if we have a laser pointer just in case. Okay, awesome. Well, hello everyone. I'm really excited to be here today. Tell you a little bit about the story and how this technology that I'm going to speak about came to be, and to share some of the research that we are conducting at the Molecular Identification Research Lab here at CFIA in Ottawa. So let's get started with this. So I want to give a little bit of an outline for the presentation today. So on the left, on this slide, you will see the very technical name of the technology we're trying to implement right now. So you might have heard, some of you might have heard of this technology, CRISPR-Cas, but of course, if you have not, you will today. I want you to focus on the right-hand side of the slide for now. Don't get into too much technicalities. So, I mean, what am I speaking about today? I'm speaking about a bacterial immune system that has been turned into a technology for modifying genomes and detecting species. So why do we care about this? Mainly because technologies that rapidly detect invasive species and plant pests are key to avoid negative effects and impacts on our agricultural and natural environment. What can we do with this? So we can develop a system that is fast, does not need heavy lab equipment, and can efficiently detect plant pests. So that's kind of like the plus of using these over other technologies that have been used before for the same purpose. So, I mean, I don't know if the mics, I'll ask you, Nicole, if the mics are open for people to speak during the presentation. If not, you can do it on the chat. I want to like open the floor and maybe ask if people know why bacteria need an immune system.
Hi, yeah, so people won't be able to unmute their screen, but if people could answer in the chat, that would be great.
Yeah,
So the
so
chat function is on the toolbar at the bottom of everyone's screen.
let's see. I just want to check. I'm not being able to actually see some of the answers. I don't know if you can tell me
Um,
if you're seeing any of the answers, Nicole.
yeah, so I maybe because you're sharing your screen, it's a but someone had said phage, Amanda Rowe, bacteria have predators too. Darlene Share has said protection. We're getting quite a few answers. So that they can fight viruses off later on.
Yeah, yeah.
Due to viruses,
Okay,
fight
great.
viruses.
Yeah, so yeah, it seems that most people got it right. So yeah, it turns out that viruses, many viruses, and someone mentioned them, the phages, infect bacteria to use their machinery to multiply and then kill the bacteria if the bacteria has no mechanism. So I do want to show you a very short video. And let me just see if I can. So yeah, so this is a T4 bacteriophage that arrives, it's gonna show that arrives to a colony of E. coli. And so after the virus recognizes the membrane of this E. coli, it binds to the host cell and it injects its genomic material. That's what you're seeing there is orange. The DNA is then transcribed into RNA, which shuts down the normal processes in the cell and directs the synthesis of viral proteins. And so some of these proteins will self-assemble to new viruses. At this point, the RNA from the virus directs the synthesis of some enzymes that rupture the cell and kill the cell, and then continue the cycle again. So one curious fact is that phages were, during the Cold War, they were starting to be used extensively in the Soviet Union to treat bacterial infections, while the Western medicine was more towards developing antibiotics. But since the breakup of the former Soviet bloc, countries began using antibiotics more and more, and so they were seen as a magic bullet, and it was clear that we moved towards antibiotics. But nowadays that we have had so many problems with antibiotics, people are moving back to study some of these phages to disrupt some of the bacteria. I find that really, really interesting. So after seeing this video, of course, we think, I mean, what do bacteria do to avoid this, or what's their immune system about, right? So for avoiding that terrible outcome of being broken into a million pieces by phages, bacteria have developed a somewhat immune system memory. So it's a little bit to what happens in humans when we're infected by a virus several times. First time the disease hits us very hard, but cells, like immune cells in our body, white blood cells, prepare antibodies so that the next time that the same virus comes, we can mark it fast and the cells target that virus and destroy it. So I'm sorry, I just jumped. When phages attack bacterial cells, something very similar happens after the genomic material of the virus enters the cell, as I was showing in the video. The bacteria has a set of enzymes called the Cas enzymes, and that's for CRISPR-associated enzymes that break the DNA, in this case, of the bacteria into pieces and incorporate some of those sections of DNA into their own genomes. And those sections are separated but by repeats. They're in a section of the genome that is separated by repeats, and that's where the CRISPR name comes from. That's cluster regularly interspaced short palindromic repeats. So I don't want to get into why this is called like this. There's a lot of repeats in between the sections of phage genome that has been incorporated. But the cool thing about this is that now that it's incorporated into the bacterial genome, they can transcribe this into RNA and use it as a way to go and mark the DNA of the phage when a new infection occurs. So we know that there's complementarity between the DNA strands, but RNA can also be complementary to these sections because now we have the same information that was captured in a previous infection. So the guide RNA recruits an enzyme, another Cas enzyme, and then marks the phage DNA, and then the enzyme cuts it into smaller pieces. So you see how this becomes the way of the bacteria to defend itself from these phages. So these two scientists, Emmanuel Charpentier and Jennifer Doudna, using cumulative knowledge of other scientists that have been studying the system since back in the 80s. So sometimes we think, oh, these scientists, of course, they're great because of what they do, but we always build on previous knowledge, right? So after they studied all this knowledge, they were studying this immune system, and they asked, what if we could engineer this bacterial system so that it could cut DNA from any organism? What happens then? So designing the system seemed pretty logical. So we saw on the previous slides how this works. So what if we could engineer guide RNAs and also, of course, produce these Cas enzymes? And then those guide RNAs would be directed to any region in the genome of any organism that we wanted to cut, right? And so if we could do this in the lab instead of just seeing it on bacteria, then we would have a system where we could cut any part of the DNA of any organism. Why is this so groundbreaking that made them deserving of the Nobel Prize back in 2020? Well, if you can direct any enzyme to cut DNA in any gene of any genome, and on any organism, you can create a mutation which then damages the gene and stops it from working. This becomes a way to know what the function of the gene is. So we as molecular biologists have this book of life written in DNA characters, but although we can kind of infer what the words mean and think about the words as genes, it is only when we disrupt these words that the real meaning becomes evident. Imagine finding a gene that we think has to do with photosynthesis, for example, in plants. What would be the best way to prove that that gene really is involved in the process? Is by disrupting the gene, right? And seeing if photosynthesis still works. If it doesn't work, then the gene is involved in the process. So usually these mutations that happen after the cut of the enzyme are a product of actually repair mechanisms of the cell. When there's a cut in DNA, there's enzymes in the cell that try to repair it, but they don't do a very good work, and they introduce mutations into that section. So usually the gene or the section is no longer functional. The second factor that shows how important this technology is that if we find a damaged gene, we can use the CRISPR-Cas system along with a repaired DNA fragment to replace the damaged DNA for good DNA. So imagine the implications that this has for disease. Many single gene mutations are related to diseases like anemia or cystic fibrosis. So there's people already working on this. Even in diseases that have multi-gene families or a diversity of genes influencing the outcome of the disease like cancer, there's things that can be done to replace some of these mutated or damaged genes with this technology. And I want to stop and show you for just a second, because I think this is really cool. Like the first time I saw it, I didn't believe it was true, but they use something called high-speed atomic force microscopy. So this video enables like real space and real-time observation of macromolecules in the nanometer scale. So you'll see here, you won't see the guide RNA because it's embedded in the enzyme, but this is the Cas RNA, and what you can see here, oh, I don't know what happened with my cursor. Can you see my cursor here? I hope you do, but that thread, brown thread that you see there is a double-stranded DNA. And so see if this works, and this is going to be just a few seconds. Hopefully, if the video goes on. Yeah, so that's the Cas enzyme on top of the DNA, and you'll see that's going to be cut on the lower part of the enzyme. The DNA is going to be cut off. That's a real image on how this actually works. And so what do we know so far? That if we have an RNA guide molecule that complements a specific section of DNA and a specific organism, the guide RNA along with the Cas enzyme will go there and cut it in a very specific way. So if we can decide that guide RNA to find species-specific DNA, fingerprints, we know that there's differences between all organisms in the planet. That's how evolution happens. The cut will only occur if the species that has the complementary sequence and not in the species that may be related but don't have that polymorphic section of the genome. So this molecular fingerprint is a consequence, of course, of our evolutionary differences, and it's imprinted in all of us. Like even two human beings are not exactly the same in their genomes, right? So to give you an example on how this works, what you see here is an alignment of the same region of the chloroplast genome of a species from amaranths. So why do I bring this up? We have been working in amaranths a lot, just because there's a few species that like Amaranthus tuberculatus and Amaranthus palmeri, they're called all-water hemp and palmer amaranth, that's kind of like their common name, that are very, very bad for crops, especially soybean and corn. And I'll talk to you a little bit more about these species, but I wanted to show you here this section of the genome of the chloroplast because everything that you see up here corresponds to different accessions from different geographical locations for Palmer amaranth. And all the species that you see down here, these sequences correspond to other species, some of which are closely related to Amaranthus palmeri. And you see there's a clear difference here. So the colors here, or the color bases, are the differences between these different regions of the genome. They're the same region of the genome, but in different species. And so if you see like this block here, it's typical only from Amaranthus palmeri and a sister species, which is called Amaranthus watsoni. So when we find those regions and in molecular diagnostics, they're great because there we can design an assay which allows us to distinguish the species. So here we actually design a guide RNA which would only match Amaranthus palmeri and as its sister species and not all the rest of the species. And we can probably use that to use the system to make cuts specifically only in Palmer amaranth. It's kind of like the example that I wanted to give you here on how this technology could be used. And so we molecular biologists have tools that we could use to detect these cuts made by CRISPR-Cas system, but there are faster and easier ways to transform that species-specific cut into something more evident and easily detectable. So CRISPR-Cas9 is the original system, mostly used for functional analysis and replacing bad sections. But scientists continue to study bacterial Cas enzymes, and they realize that this is a big family of enzymes that somehow have similar functions, but some of them can do things a little differently. So what I'm showing here is a Cas12 enzyme, not a Cas9 enzyme, which is, they do pretty much the same. They can find a target, they can cut it, but Cas12, when it finds the target, it cuts the target DNA, but all of a sudden it goes on a cutting frenzy, cutting any other free-floating DNA that is on its vicinity. And this activity is known as collateral activity. I can use this characteristic of the Cas12 enzyme to add a free-floating piece of DNA that has a fluorescent probe, a dye that will fluoresce, and a quencher. This is an inhibitor that when it's closely located to the fluorescent dye, it won't allow the fluorescence to occur. But if we can cut and distance the inhibitor from the fluorescent dye, then we will have fluorescence in our tube where we produce our chemical reaction. So a workflow to detect a specific species would look something like this. You have an amaranth plant or its seeds, you extract DNA. To increase the specificity, you could amplify the region where you are supposed to find the polymorphism, that means of PCR or other technologies. And then you create your enzymatic reaction where you mix your enzyme, your guide RNA, and your target DNA. And if you find the species that you're supposed to find or you're expecting, then you'll see fluorescence, and you just need UV or blue light for doing that. And so now you can tell me, why does this matter? Can't we just look at the plants? And I mean, tell me what it is. Like taxonomy has been used for hundreds of years to distinguish plants. So why don't we just go and look at the plants. Well, not always, and not for all stages of plants, this is straightforward. So I'll throw out another question, if you can answer to me in the chat, what difference you see between the seeds in the left and what the seeds on the right look like? Is there any differences that are evident for you? And then I'll give you a few seconds, and maybe if Nicole can tell me if anyone has any idea what the differences are.
Someone typed in the chat, nothing hard to tell.
Hard to tell, yeah. Yeah, this is hard to tell. These are actually two species of amaranths. The one in the left is called Amaranthus powelli, and the one in the right is Amaranthus retroflexus. So sometimes even for seed analysts, this is really hard to distinguish. Also, plants have a lot of plasticity, so seeds, even sizes of seeds, can change due to environmental conditions, climatic conditions, etc. So seed analysts sometimes need to look sometimes at 1,000 seeds to take one of these contaminant seeds, if it's regulated, for example, try to differentiate them and classify them. Are they regulated? Are they not regulated? If they're regulated, this may become a problem in certification on trade. The same manner, farmers have weed seeds in their soils that cannot be easily identified. They have to sometimes send them to private labs. I tell them, please grow my seeds. Sometimes you have to test them for herbicide resistance because I don't know what's going to pop up in my crop this year. But that sometimes takes weeks or months, and that stops taking in-season management decisions. So, for example, what pre-emergence herbicides should I be using this year, depending on what species I have, and what herbicides can I use this year, depending on the crop I'm growing, right? But that creates an issue. So in the agency I work for, the Canadian Food Inspection Agency, we regulate the import, export, movement of plants, seeds, plant products. We deal especially with the species of regulatory concern, but other species that may impact farmers. We also deal with those. So we know that invasive plant species introduced into the Canadian natural and agricultural environments can result in detrimental effects through competition with native species, changes in soil composition, because they also bring their own microbiome along with them, and they have specific chemicals that they produce. They can hybridize with some of the native species, and that also creates a problem. They can reduce local biodiversity because usually they're very competitive. They can take over. They can introduce pathogens, and some of them can even be poisonous, and so they can be harmful for human health. So, I mean, we can address the problem of invasive species in different levels, but this is kind of like a graph that we always show. It's like prevention is first, eradication, containment, and long-term management. So, as you see from the graph here, the area infested, which is here, and the time that it takes to control this, and the cost of controlling the pest, they grow exponentially. Right? And so if we don't address the problem in its early stages, that becomes like a bigger problem to manage in all senses. See that the bulk of what's invested was 2.2 billion annually to control invasive species is dedicated mainly to management. So we're trying to move away from always trying to do management and do a lot of prevention. So part of this prevention can be done through early detection, and therefore that's where molecular technologies come into play. And so while we at CFIA and federal level, we regulate many species, I want to go back a little bit and talk about specifically Amaranthus palmeri, which is already regulated in several states in the US, and it has a big impact. You can see here millions of dollars, even billions of dollars back from 2000. This is numbers from 2015. This is just how bad the production of these main crops was affected. And although Amaranthus palmeri has not been like fully detected as populations in Canada, we already have detected some plants in Ontario and Manitoba, so they're coming. And Amaranthus tuberculatus, which is all water hemp, is already widespread in Canada. So this is a real problem that we have now. And so I'll give you an example here. This is a picture of corn, a cornfield in Nebraska, and all in between you see plants of Amaranthus palmeri, right? So one single plant under ideal conditions can produce up to 1 million seeds. resistant to multiple herbicides, which become now harder to control. We're running out of options right now. And the seeds are very difficult to distinguish. So if we have them mixed in like a trade commodity, that's also going to be a problem. Even like full grown plants sometimes are difficult to distinguish from close relatives. So you need flowering usually, but you cannot really wait until they flower, then it's too late for you. This plant, of course, as I was saying, has implications in trade. So if you heard the news, and I know that there's a lot of political background behind this news, but in 2019, China suspended two big Canadian companies from Manitoba and Saskatchewan, Viterra and Richardson, alleging they had detected quarantine pests in canola shipments. So their official communications said something like, China Customs Administration said in a statement posted this day that 4 custom offices in China found pests, including this is a series of bacteria, fungi, but also French wild oat, and here is Amaranthus palmeri. There was like, even though inspectors here said we didn't have that, there were no additional technologies to say, okay, yeah, we can do another test, we can do a molecular test and show that that's not true. There's a lot of things that go around this, like China wanting to promote their own agricultural rapeseed and stuff like that. There's also the political thing that happened that year, but either way, the canola industry contributes almost $27 billion and 250,000 jobs to the Canadian economy. And in 2018, we exported 11 billion worth of canola to 50 markets. If you see here, 3 billion, almost 3 billion were exported to China. So China is Canada's biggest canola exporter. And you see on those 3 years after the 18, how we had to kind of rebound, because this is the biggest market that we have for canola. And for China, like over 90% of everything that's rapeseed oil derived from canola comes from Canada. Like it's a very small market that comes from other countries. So this affected, of course, China, but mainly our economy. So it's evident that, I mean, the implications of not detecting these plants on trade or agricultural environments has devastating consequences. And since, I mean, I work in the development of molecular technology, that's what we do here in this lab. I'm a true believer that by applying these technologies, we can be more efficient in detecting these invasive and weedy plants by implementing some of these methods. So I'm going to go back now to explain to you a little bit of the technology, explain of the impact of these plants on trade. So I want to show you a little bit of the result that we have had using this technology. There was a lot of work that went into this. Like we have been working on this for the better part of last year, but for the ones that have done a little bit of work in molecular biology, they know that it's missing 90% of the times and hitting 10% of the time. So there goes a lot into standardization, and for that I have to thank Andrea Dupras, who's the main technician on the lab. She has been working hard at this. But what I want to show you here is that we designed a guide RNA that would complement DNA exclusively from palmeri and its sister species, that's Amaranthus watsoni. So to see if we could identify it using this technology. So the test that's shown here shows specificity after 60 minutes of reaction time among 17 different species. And if you can see here, you will only see the acronyms for each one of the species, and there's several accessions per each one of the species, so several samples. But what matters here is that samples 32 to 38 APAL correspond to Amaranthus palmeri, and 64 here corresponds to Amaranthus watsoni. And if you see, you can only see fluorescence on those tubes corresponding to palmeri or watsoni. So this test was quite specific and quite efficient in detecting these two species in a very short amount of time. Now, we have further extended the development of this technology to detect plant pathogens. So we have obtained DNA from one of our collaborators and our research here at CFIA, Dr. Guillaume Villedon, who's studying the causal agent of sudden oak death, Phytophthora ramorum, a disease that kills oak and other tree species and has had devastating consequences in the tree population, California, Oregon, but as well as some parts of Europe. So some of the symptoms that you can see there on a tree trunk in the background include cankers on the tree trunk and dieback of the foliage. Many cases, the tree cannot take it and it just dies. So on this test here, we took a guide RNA that is specific to Phytophthora ramorum, which is number 7 here, but all these isolates, 7 to 16, are actually from Phytophthora ramorum from different lineages. And these other 3 species that you see on the top are 3 very closely related species. So for a test like this to work, you have to be able to distinguish your target species from the closest related species. And so this work was done by a student from Carleton, who's no longer in the lab, but she did an excellent job in standardizing this. And as you see, by 10 minutes, you can only see samples 9 to 16 lighting up, and then 20 and 30 minutes, with the exception of those 2 samples, 13 and 14. We did a little bit on these 2 samples. They come from isolates from Europe, which I've been told from Guillaume that they are distantly related to the other isolates. So we did a little digging and we investigated some of the sequences of these isolates that are on the GenBank database. It turns out that the region where our guide RNA binds, it's highly mutated, highly different in those 2 isolates precisely. So that's why we wouldn't see fluorescence there. But we continue to work in designing an assay that can distinguish all the isolates from Phytophthora ramorum, or we could also create an assay specifically for that lineage. So as we continue to test new species, we're also investing our efforts in increasing the efficiency and ease of use of this technology. So one of the most amazing things, and I get really excited about using CRISPR technology because I see all the possibilities that it has for doing molecular diagnostics, so not only functional biology. So right now, what we're trying to do is we are using a buffer which allows you to extract DNA in less than 20 minutes, something that you could potentially take to the field. And then after that, once you have a little bit of that DNA, you can do something called an isothermal reaction. So if some of you have heard of PCR, we usually need an equipment, a thermocycler that increases and decreases temperature and goes through several cycles to amplify a region of DNA. Instead of that, we are using a recombinase enzyme, which can be used at room temperature. Although the ideal temperature might be closer to 37 degrees, you can do it at room temperature and it still works, amplifies that region. And then the Cas enzyme, again, ideal temperature is on the 30 degree or past 30 degrees, but you can do it at room temperature and it will still find its target. It will still cut the probe and it still will produce fluorescence. So once we have all this standardized, most of it is standardized already. We're just testing the DNA extraction buffer, but you can do all this reaction on a bench without heavy equipment in around an hour. And you could potentially implement this for almost any organism. It has been tested before with COVID-19. It has been done in some pathogens, but as long as you are able to find the right sections of polymorphisms or changes between sequences, you can do it to identify specific species. So our idea is to implement this without no heavy equipment, eventually a field test, and later on, try to turn it into a strip assay, similar to the strip assays that we took on when we had the pandemic. So it can be turned into something very similar to that. So just to wrap up, I'm not going to extend too much more, but other plans we're working on here in the lab to generate molecular technologies, that being CRISPR or other technologies. We are studying wild mustard, as you see there on the left, toad flags, wild oats, daughters, that's a cuscura, that's a parasitic plant, which is, I find it fascinating that through evolution, it has lost some of the genes from the chloroplast genome, because now what it does is basically sucks the nutrients out of the plants that it colonizes or that it parasitizes. And yeah, for most of these plants, we are developing specific barcodes. Down here is jointed goldgrass. I missed that one. But we're developing specific barcodes that allow us to identify them directly from the soil seed bank or pick up contaminants in trade shipments using, CRISPR is one technology, but we also use metabarcoding we use a lot of bioinformatics. And so, I mean, for those of you who have not heard about metabolic coding, is very similar to what you hear sometimes about environmental DNA, right? Like picking up environmental DNA and looking at one region that can distinguish all these plants. For wild oats, we are actually doing a project right now to study mechanisms of herbicide resistance. There's a big problem with wild oat, especially in the prairies, and developing markers that would also rapidly allow to identify some of the populations. So those different populations of the same species have different levels of herbicide resistance, and that's a big issue for farmers nowadays. And when for amaranthus, again, we are doing the CRISPR assays, but we are also studying variation at the whole genome level and at the chloroplast genome level using a series of bioinformatic tools. So with that, I just want to finish up by acknowledging some of the people that have done the work. I mean, the people from my lab here. This picture was taken just a few days ago. That's Sangi, who's a grad student, Noah, an undergrad student, Jacob, also a grad student, Jillian, undergrad, Andrian, who's a technician. But Andrian and Rina, who's not in the picture, did most of the work for CRISPR. The pathogen identification research lab led by Dr. Guillaume Galindo and Isaac Firth, who provided some of the material for the phytophthora experiments. Dr. Roy Wang, who's in Saskatoon with CFIA, she has provided financial support and some of the material for seed work with amaranthus, and most of the finding that comes from the Canadian Food Inspection Agency. And there's some pictures here of the past members of the lab who also have provided a lot of help to build up the lab. So yeah, with that, I want to finish and thank you for listening, and I'll take any questions.
Thank you so much, Dr. Galindo González. We have quite a few questions in the chat, in the Q&A box below, so I can start reading those out right now.
Sure.
First question by David Latour. If you get a shipment of grain with possible contaminants, can you just take a sample and extract DNA from all seeds together and use different probes with different fluorescence to do them all at once?
Yes, there is a way. There is a way to use different fluorescent probes to do some throughput, although this technology is not specifically designed for high throughput. In the case of having a mix of potential contaminants, the technology that I would go with, if I don't have a specific target, but I'm just looking to see if I have different species that might be different weeds, I would go with something more like metabar coding, next generation sequencing, because that allows you to know all the potential contaminants, even the ones that may not be a target. So they might be other seeds that are not a target, but they still may be detrimental in trait. So in that sense, I would try to go with something like next generation sequencing. It would require for CRISPR to do a more throughput analysis to do try to separate the seeds first. And then there is systems like Fluidigm with microfluidics that can be used to test all the potential specific guide RNAs and mix it with all potential samples. That's kind of like what they did before with viruses. So it would be a similar approach, but if you really want to see like all the diversity on something that's mixed, and you're not targeting like a single species, I would go with something like next generation sequencing. If you're targeting just one species, then this approach would be good to do.
Thanks, Leo. David also asked, can you use the same technology to screen for herbicide-resistant invasive species?
Yeah. So yeah, David is actually a good friend of mine. Yeah, you can. That's one of the things that we have thought about. So there's two types of herbicide resistance. There's the target site resistance and non-target site resistance. For the non-target site resistance, really difficult because that's more of a multi-gene metabolic process of the plant. But for many of the interactions where there's only one gene and potentially a few mutations in that gene. So if we talk about acetyl-CoA carboxylase or acetolactase. Forget the last name of this enzyme, but these are two of the main groups of the main proteins that are affected by herbicides in some of the populations right now. And you find mutations that are associated specifically with the resistance to certain groups of herbicides. So for those ones, if you find a mutation, arguably you could pretty much standardize a test like this. You just have to make sure that the mutation is related to the phenotype. So do some phenotypic testing, and after you could, in theory, you could do it. Yeah.
Thank you. So just reading from the Q&A box. Next question, could this technique be used to sample eDNA for a target species and get results in situ?
Yeah, so I think that comes back to the question before from David. If we have a single target species, we could do it. And the good thing with this technology, actually, we are just trying to single out one single species, is that the pre-amplification step that you have allows you to create more molecules of that region where your polymorphism is supposed to be. So you have two levels. You have a level of amplification of that region, and then you have your fluorescent reaction. So the times that this has been used before for increasing resolution. So if you have a very diluted contaminant or a very diluted species that you want to target, you can get to the level of attomolar molecules, so very small amounts of molecules. So once we develop this, you have to standardize it for each one of the species, of course, but it has been done before for viruses, so I believe it's possible for other species.
Great. Next question, have you tested the tool in low concentrations of DNA by simply extracting from plant material directly?
Yeah, thank you for that question, Valerie. No, we haven't, but we want to do it. The increasing resolution of this technology is based on that pre-amplification step, but there's ways that you can modify the technology to generate higher levels of fluorescence by changing some of the tail sequences that you put in some of the mixed components there, like the guide RNA. And so you could arguably get rid of the amplification step and try to go to lower concentrations by increasing the level of fluorescence. I haven't done it, but that's something that I have in mind that eventually we want to do.
Valerie also asked, is the Cas12 enzyme or the isothermal recombinase highly affected by chymic acids and other inhibitors?
I have no idea. I have no idea. So far, we just tested it on amaranthus, where it doesn't seem to be a problem. I would have to look into, we're going to try to test it on other plants. I don't know what the chemical composition of either like seeds or plant materials would be in that sense, but that's I think a really interesting point. As we move forward, we will see if it affects, depending on the tissue, stage of development or type of tissue. That problem does not seem to, of course, arrive with material that comes from pathogens. As long as you can get the DNA, we don't have any cleanup stages though. So far so good, but we'll see in the future.
Thank you, Valerie, for your questions. Jeff Barnes has asked, how is this technology different from or better than LAMP technology, which stands for loop-mediated isothermal amplification?
Yeah, this is, I'm gonna answer from theory because I haven't worked with LAMP, but what I've seen from LAMP in the past is that sometimes the design of the experiment itself, primers, conditions for getting to an ideal amplification are a little more complicated than this. This is recombinase primers pretty much, work easily, you don't have to standardize a lot. Sometimes even the PCR primers work with their combination. There's not much issue with this. The rate of reaction, not sure about it compared to enzymes that are using LAMP. But it seems to be the design itself, it seems to be pretty straightforward and a little easier than LAMP.
Thank you, Jeff, for your question. A lot of amazing interest in your research, lots of great questions. We'll just go with a couple more in interest of time. I'll move to the chat now because I see there's some questions in the chat box. Comments, a lot of great comments thanking you for your work, Leo, and the excellent work of your team. I see a question by Anne Eastman. Are you limited by IP for making a commercially available kit?
Yes, I'd like be working for the government. I wouldn't be the one designing, but we do when we have a methodology that's standardized. Usually, our main clients are labs that do diagnostics within the agency. So we have research labs and we have diagnostics labs. Ours is a research lab, so we develop technologies and then we try to validate them as much as we can. And then diagnostics creates panels and they test them for efficiency in detecting whatever target we're aiming for. We also have, sometimes we do agreements with private companies where we, if we see that this might be able to get out into the market, then they say, oh, this is the technology that we want to adopt. And so we transfer the technology and they are the ones who commercialize the technology. At this point, I'm concerned just to develop a good solid assay that can be used either in the lab or outside in the field. But eventually, we've seen that before, that private companies have sometimes interest in developing or standardizing and commercializing the assay furthermore.
Thank you. Jeanette Amangian asked, first she says first they say thank you very much for your excellent work and asked if it would be possible to send us a list of invasive plants that you are working on.
Sure. If you want, there's my email. I don't know if is it still being shared, my screen?
Your screen is being shared, yes. Is it?
Yeah.
I can also type, if that's okay, Leo, your
Yeah, that's
email
okay.
in the
So,
chat.
because I don't know where the question is coming from, but just, I mean, write me an email and I can send you a list of the plants that we're working with right now. Yeah, for sure.
I'll just write your email in the chat. Janet also asked if we will get a copy of the presentation. I will say that this is being recorded and it will be available soon on the Invasive Species Center website and also on our YouTube channel.
Yep. Okay.
Okay, thank you so much. I'll see if there's any other questions. Okay, I don't see any further questions just in the chat. Many people thanking you, Leo, for your presentation, for your very interesting presentation and sharing with all of us. I will close off then with I'll start sharing my presentation again. Okay. Okay, well, thank you again, Dr. Galindo González, for presenting today. It was extremely informative learning about yours and your team's research, and we really do value your time and expertise in invasive species identification and management. We'd also like to thank you all for tuning in today and for your amazing questions. And again, this webinar was recorded and will be posted on our website at www.invasivespeciescenter.ca. And just a reminder as well to please take a couple of minutes to fill out our survey. We would really appreciate it. Our next webinar will be tomorrow, September 19th at 11 a.m. as well, titled Sampling Efforts Required to Detect and Remove Asian Carps During Response Activities in the Laurentian Great Lakes Basin, which you can register for our website as well at invasivespeciescenter.ca/webinar series. And thank you so much, everyone, for